Red light therapy has been reported to have many health benefits, one of them being the reduction of oxidative stress that occurs from a buildup of free radicals in the body. And since this is a hallmark of many complex chronic inflammatory conditions, including infection associated chronic conditions like Pandas PANS, chronic Lyme, post infectious ME, long COVID, even autoimmune conditions and neurodegenerative diseases, I'm going to explain in this video the science behind why red light therapy might heal your mitochondria. However, nothing you see or hear in this video is intended to be medical advice. This is for educational and entertainment purposes only. So let's dive into the video. In order to understand the biology behind near-infrared light and how it interacts with your mitochondria to reduce oxidative stress, we need to understand how the mitochondria work. So you can think of your mitochondria as a kind of a hydroelectric dam. The hydroelectric dam works because you have a very large number of water molecules on one side of the dam, creating a lot of pressure. So you have high pressure on one side of the dam's wall and you have low pressure on the other side. And so once this control gate opens, water flows through and it spins a turbine to generate electricity. And so keep this in mind as I talk about the mitochondria, because they kind of work in a similar way. There's some parallels here. So this is a very busy slide, I'll admit, but let me walk you through it step by step. And I think here in a few minutes you'll understand that this isn't that scary. This isn't that intimidating. Okay, so let's just focus on this top right part of the slide, looking at the mitochondria. So if you didn't know already, this is the characteristic mitochondria appearance, where you have this internal maze-like structure in the middle of the mitochondria. And you can see that there are two of these burgundy lines here, these dark red lines. This represents the two different plasma membranes that are found in the mitochondria. So you have an outer mitochondrial membrane, and then you also have an inner mitochondrial membrane. And the inner mitochondrial membrane has a much larger surface area than the outer mitochondrial membrane, which is what gives it this characteristic appearance. And so when you zoom in on a specific region of the mitochondria, you can see that there are a large number of protein complexes that are densely packed in the inner mitochondrial membrane. And we're going to go back to this hydroelectric dam analogy, and the inner mitochondrial membrane will be, I want you to think of that as the dam wall. And so there are these protein complexes that are embedded in the wall of this dam, okay, so to speak, the inner mitochondrial membrane. And what they do is they pump hydrogen ions from one side to the other. And what this does is this creates a gradient of hydrogen ions where you have a large number in this space called the intermembrane space, which is the space between both of these plasma membranes. And you get this buildup of hydrogen ions, or this hydrogen ion gradient. And similar to how water flows through that tunnel to spin the electric turbine to generate electric power, the hydrogen ions also flow through this molecule called, or this protein called ATP synthase. So think of this as the dam of, think of this as the turbine. And when these hydrogen ions flow through ATP synthase, it allows this enzyme to catalyze a reaction. And what it does is it takes ADP, and ADP stands for adenosine diphosphate. So the D stands for di meaning two phosphate atoms, two phosphate groups or attached to adenosine. And it adds a third phosphate to the ADP molecule, creating ATP, or adenosine triphosphate. And adenosine triphosphate is considered the energy currency of your cells, because there are many enzymes inside the cell that use ATP to power enzymatic reactions. Okay. Now, the question is, how are these, hydrogen ion pumps here that are embedded in the inner mitochondrial membrane, how are they pumping these hydrogen ions and creating this hydrogen ion gradient? And the answer is something called the electron transport chain. Okay, so just as electrons flow through a copper wire to allow you to flip on a light switch or turn on your coffee maker, electrons flow through these protein pumps or these hydrogen ion pumps, and the flow of these electrons actually powers these pumps, and it allows them to pump hydrogen ions from here to here. And so the reason this works is because these protein complexes contain a large number of iron sulfur clusters. So these I'm representing it as though they're spaced out, but they're actually very densely packed together, and they contain a lot of iron and a lot of sulfur that's clustered together. And so the electrons flow through the iron sulfur clusters, and this energy that is released as the electrons flow through is enough energy to change the shape slightly of these protein complexes to allow the hydrogen ions to flow through to the other side. Now, the flow of these electrons through this electron transport chain has a very clear starting point and a very clear ending point. Okay, so let's start at the starting line and understand where these electrons are coming from, because this is very important. So when you eat things from your diet, such as sugars or fats, they will enter into metabolic pathways into the cell. So sugar will be transported into your cells. It will flow through a linear metabolic pathway, and it will be converted ultimately into a molecule called acetyl-CoA. Fats do the same thing. But they flow through a different metabolic pathway. But eventually they are converted into acetyl-CoA, which enters into a circular metabolic pathway inside the mitochondria, specifically right in here, in this space called the mitochondrial matrix, this light pink area. And the acetyl-CoA will be converted into a number of different metabolites. And as this occurs, a molecule called NAD will be produced. And NAD is oxidized, which means that this hydrogen ion is removed, and the electrons that hold the bond together enter into this complex 1. And that is the source of the electrons. It's from your diet, basically. So these electrons flow, and they have a clear ending point, which is this big protein complex here, this purple protein complex, is called cytochrome C oxidase. And what it does is it takes the electrons, combines it with oxygen and hydrogen to make a water molecule. And this is why we breathe oxygen. This is why we need oxygen to survive. It's because oxygen acts as a terminal electron acceptor in the electron transport chain in your mitochondria. So without oxygen, these electrons cannot be absorbed anywhere, and they'll start reacting with other molecules. And this is not a spontaneous reaction. It's catalyzed by this enzyme here, cytochrome C oxidase. Okay, so next we will talk about reactive oxygen species, because reactive oxygen is a byproduct. It's an unintended consequence of premature exit of these electrons from the electron transport chain. So think of the electron transport chain as a roller coaster ride. If the cart of the roller coaster, or in this analogy, which would be these electrons, if they come off the tracks, so to speak, there could be some really bad collateral damage that could occur, right? And so premature exit of these electrons from this flowing electron transport chain, what will happen is the electrons will spontaneously react with oxygen, and that will form a number of different species of free radicals called reactive oxygen species. And usually they prematurely exit this electron transport chain at this site here, complex 1 and this is actually complex 3. And this will, again, react spontaneously with oxygen, creating ROS, reactive oxygen species, which is a type of free radical. And these free radicals will then collide with nearby molecules, and they will strip electrons, or they will donate electrons to these molecules, creating another free radical that then goes and collides with another molecule to create another free radical, and so on and so forth. And it creates this cascade of free radicals that can ultimately cause a lot of damage inside your cell. There is another molecule called nitric oxide, NO. And nitric oxide likes to bind right here on this purple protein called cytochrome C oxidase. And this will gum up the electron transport chain. So this will prevent the electrons from efficiently flowing all the way through and allowing the enzyme here to catalyze the reaction, which is this reaction here in brackets, which is basically electrons plus oxygen and hydrogen ions to make water. And so if a nitric oxide molecule sits right here in cytochrome C oxidase, you will get this premature exit of the electrons from this electron transport chain. And these electrons, as I mentioned, will react with oxygen and other molecules to create free radicals. So this is obviously not good, not a good thing. So here's where the therapeutic benefits, or at least one of the benefits of near-infrared light come into play. Because it's been reported that near-infrared light, when it shines in your tissue, it will be absorbed by this purple protein, the cytochrome C oxidase. And what it will do is it will displace this NO molecule, and it will free up the electron transport chain, allowing this enzyme, cytochrome C oxidase, to do its job, to catalyze this reaction here. So again, when nitric oxide is displaced by the near-infrared light, the electron transport chain will improve in efficiency. And what this does is it also improves the ability for these proteins to pump the hydrogen ions to this side of the mitochondria, the intermembrane space, to improve the flow of the hydrogen ions through ATP synthase to make more ATP. So not only does it reduce the oxidative stress because you're making fewer reactive oxygen species, right, from these complexes and even from here, but you're also increasing the amount of ATP that is produced by the mitochondria. So these are the benefits, at least one way in which the near-infrared light is reported to improve the bioenergetics of your mitochondria. And so now the question is, where does this nitric oxide molecule come from? I mean, if it's so detrimental to the function of your mitochondria, then where is it coming from and why are we making it in the first place? And so the answer to that, or at least one of the sources of this nitric oxide, is from your immune system, from inflammation. Okay, so nitric oxide can be made when you encounter certain pathogens, right? Viruses, bacteria, fungi. There are immune cells that will make an enzyme called inducible nitric oxide synthase, or INOS. And INOS is triggered, or the production of INOS is stimulated when these immune cells encounter certain triggers from microbial products, such as lipopolysaccharide, which is from gram-negative bacteria, and this is the strongest inducer of INOS production. But peptidic glycan, which is from gram-positive bacteria, can also stimulate INOS production, as well as viral double-stranded RNA through TLR3 will be stimulated by double-stranded RNA to induce the expression of this enzyme INOS, as well as fungal beta-glucans, such as dectin-1, will make INOS. And again, INOS is the enzyme that makes nitric oxide. And it makes it in a very robust, potent manner. Because there's other nitric oxide synthases that are not inducible, that are constitutively expressed, that will make nitric oxide. But this one will crank out a ton of nitric oxide when its activation is when its gene expression is turned on. But there's also pro-inflammatory cytokines that can stimulate the expression of INOS. So it's not just infection from pathogens. Chronic inflammation can also stimulate INOS expression if these pro-inflammatory cytokines are too high. And of note too, INOS expression can be suppressed by anti-inflammatory signals or anti-inflammatory cytokines such as IL-4, IL-10, and TGF-beta. And glucocorticoids or steroids can also reduce the amount of INOS expression to reduce the amount of nitric oxide in the body. Now, if nitric oxide is harmful, then why are we making it? And the reason is because nitric oxide is not only toxic to our own cells in very high doses, but it's also very toxic to pathogens. So this is a part of your innate immune response to infections. Typically say that you, the listener, are currently suffering from mitochondrial dysfunction. Now let's do a quick thought exercise and try to understand what is going to happen if you subject yourself to any type of red light therapy. So as we mentioned earlier, the red light therapy, specifically the near infrared light wavelengths, will deeply penetrate your tissue and touch nearly every mitochondria. And this light will kick off the NO, and that NO will actually be free to react with things around it. And so one of the things that nitric oxide will spontaneously react with is another reactive oxygen species called superoxide. And superoxide is basically it's an oxygen molecule with an additional electron. And so nitric oxide plus superoxide free radical will form another very potent free radical called peroxynitrite. And peroxynitrite will damage a lot of things inside of your cell. And so one of the things that could occur is you get damage of not only nearby lipids, but also these hydrogen ion pumps. If you damage these, that will also contribute to the mitochondrial dysfunction. So that's something that you obviously want to avoid. So you have to be careful if you do decide to sit down in front of a red light therapy lamp, and because you're going to potentially be forming a lot of peroxynitrite. And so, again, I cannot give medical advice, and this is not medical advice at all, but I will say that if it was me that had mitochondrial dysfunction, what I would do was take a very high quality supplement like NAC, and I would take that about an hour before I sit in front of a red light therapy lamp, and I would take the NAC on an empty stomach so that it increases bioavailability, and hopefully that NAC does its job and it builds up the pool of glutathione inside of my mitochondria. And glutathione is a natural antioxidant that's inside the mitochondria to neutralize reactive oxygen so that I could prevent peroxynitrite from forming. But again, that's not medical advice. Take it for what it's worth. But with that, I thank you for listening, and I hope you learned something. And we will talk to you next time.